Well, hi everyone, and welcome to this video on how to organise your data for Eagle. So the next couple of slides, I just want to talk you through the input files themselves and what they need to look like. Before I do that though, let's talk a little bit about the types of study populations that Eagle can handle. So be it plant, animal or human types of GWAS, Eagle can deal with all of those scenarios. Eagle will handle data from inbred or outbred individuals, and also the populations can be structured or unstructured or even semi-structured. So what I mean by that is if you're sitting in, say, the animal and the plant space where, I don't know, if you're doing experimental crosses such as F1s or F2s or collaborative crosses or whatever you're into, then Eagle can deal with that type of data. Or if you're coming out from the human space where obviously your control over your crosses is not there, things are a little bit more unstructured, then Eagle's appropriate for both those cases. You don't even need to know the pedigree. Eagle obviously will go back a step. Pedigree is very important for analysis, so you need to account for that within your analysis. But what we do is we will estimate relatedness from the SNP data that we have. So in this slide, we just have a summary of the different types of data file that Eagle's expecting and their structure or their attributes. In saying that, it is dependent upon the design or the type of analysis that you're running, whether you need all these data files. So for example, if you're coming out of the human space where each individual for a trait has a single measurement, then you won't need the Z matrix. But if you're doing, say, an animal or a plant study where for a trait you might have multiple measurements, then you will need the Z matrix. Also, the map is optional. So, it's hard to believe that you wouldn't have a map in this day and age, but if you don't, then you can still use Eagle. Obviously it makes the interpretation of the results a little bit difficult, but Eagle doesn't actually require the map to be known for it to do its analyses. So you can read this table as well as I can, so I won't go through every entry. That will just put you to sleep if you're not already asleep, but there are a couple of things that I will point out. So first of all, of the marker data files, we can handle p-link ped files or plain ASCII text files. Please don't include column headings in that file, and also in terms of row headings, don't include row headings in any of the files. Row headings are out. If you have row headings in the file, that's just going to cause us havoc. I suppose the only other thing I'll say is in terms of missing data, yes, we can tolerate a certain amount of missing data, because obviously missing data in real studies are just a fact of life. But make sure you don't have any missing data in that Z matrix. So with the marker data file, we are expecting SNP genotypes, but that's not to say you can't handle other types of markers as well. So there are ways of making dominant markers and highly polymorphic markers SNP-like. So as long as you can do that, then we can handle that situation. In terms of the data file itself, then we expect the rows to correspond to different individuals and the columns to code up to different SNPs. If you're using a p-link ped file, then that format's very set, and you just go to the website and we follow the same rules as you would for a normal p-link ped file. But if you have a plain text file, then you do have a little bit of more flexibility. So you still need to have a space separated file where the rows are individuals and the columns are the SNPs. But in terms of how you code the actual SNP genotypes up, that's largely up to you. So what I've done is I've just given you a couple of examples, and of those three files that I've shown you, they're actually all equivalent. So even though they might look very different, they're actually equivalent ways of coding up the same information. So in that first file, that first block of text, then I'm just using zeros, ones and twos, so not very imaginative. But in this second block, here I have the allelic information. So as long as you don't have any spaces between those alleles, everything is fine. And then in this third case, which is a little bit abstract, I'm actually coding them up as Tom, Dick and Harry. So the only requirement is just to make sure that you don't change your coding halfway along. So you need to be consistent. What I mean by that is if you start coding, say, the first half of your SNPs on the genome as Tom, Dick and Harry, then you can't change your mind halfway along and start calling them Larry, Curly and Moe. That's not going to work. So as long as you're consistent, things are good. Now we do separate out the marker data and the phenotypic data. And in the phenotypic data, again, the rows correspond to individuals, but you can have multiple rows on the same individual, and that's going to be dependent upon your design. The file can contain multiple traits, so you don't need to have a separate file for each of your traits. You can have it all contained within this single file, and the analysis works best if you're dealing with continuous traits, but it can handle binary traits. So what I mean by that is the underlying models are linear mixed models, and linear mixed models are geared towards the analysis of data that's normally distributed, but linear mixed models are fairly robust, so you can deviate from that. And I've seen analyses, and we've done analyses ourselves, where we've actually had binary traits and it's performed reasonably well. The only price you pay is maybe you lose a little bit of power, so maybe you lose the ability to find some really small results if you're dealing with a binary trait. But otherwise, preferably we'd like to deal with continuous traits. So what I have there is just an example of actually some real data that we looked at within our paper, and it's just a snapshot, so it's not everything, it's just really just a condensed version, just to give you an idea. So you can be pretty flexible as long as you just have spaces, and make sure that you don't have a space at the beginning of the line, because that's going to cause us troubles. And also don't have spaces between the titles. So what I have there, that first row are the column headings. Make sure you don't have crazy characters and spaces between your names. So that's why you see in the examples I've actually used an underscore to denote a space. So the map file, I think, is pretty standard in its structure. We do have the requirement that the rows of that map file have to correspond to the ordering that you have within the marker file, and we can actually check that. What we will do within Eagle is make sure that the number of rows you have in the map file is the same as the number of columns you have in your marker file, but we can't check the ordering, so we are reliant upon you to get that right. The only other requirement of this file is that, well, it's up to you whether you have column headings, but you need to make sure that the first column corresponds to the SNP names, the second column contains the chromosome labels, and the third column contains the position of that SNP on that chromosome. So the units, we don't care about the units, so you can have whatever units you like for the position. You just have to make sure that those three columns are ordered in that way. And then the last type of file I'll talk about is this Z matrix. So that is probably linear algebra speak, in that the Z matrix is really just a design matrix or incidence matrix that allows us to match the genotype to phenotype values when there are multiple values for a trait. So I'm not talking about the situation where there are multiple traits. I'm actually talking about the situation where there's a single trait, but you might have multiple measurements for an individual. So for example, say you're in a plant study and you're running a field trial where you have a number of varieties. Then what you would do is, for each of those varieties, you'd run them across a number of different plots within your field. So you will have multiple measurements being recorded under the same genotype, if you like. So what I've done is I've just given an example of, given that incidence matrix of ones and zeros, an incidence matrix or the Z matrix can only contain ones and zeros. If you have any other value, then Eagle will have a bit of spaz. But given that you've got a matrix that has ones and zeros, then what I'm doing there is just showing you how that maps the phenotype to the genotype. So in this particular example, the first two rows of phenotype actually refer to the same individual, and that individual's genotype is in the first row of that marker file. For the third row of that Z matrix, what that's telling us is that the third row of the phenotype actually corresponds to the third row corresponds to actually the third row in the genotype file, and so on. And you can work your way through and you can see how things are mapped. So my apologies, that has taken longer than I thought. If you're still stuck, if you're still having troubles, then do have a look at our website. So my guess is that you have gone to our website even to find this video, even though the video is available on YouTube. But if you haven't, then go to our website and have a look. There is information there on quick start guides, and we've got other videos such as a tutorial and so on that hopefully, if you're still struggling to get data into Eagle, then that will get you on your way. If all else fails, email us. So email us at eaglehelp@siro.au, and we will do our best to get you up and running. So thank you very much.